Hi, I'm Hayato Anzawa. I talk about our study, large-scale assessment of ChIP-seq quality metrics toward peak call-free quality control. So first of all, I introduce about VSN, the new metrics for quality control for ChIP-seq experiments. So strand cross-correlation is a correlation function between the distribution of the depth of forward and reverse reads. So for one binding event, there's two peaks can be observed. So one is a forward read of the read depth, and second is reverse read of read depth. And we can calculate the correlation between these two depth distribution, and it has a single maximum value. And recent study, the maximum value of cross correlation coefficient can be written as a following equation. So here, large M means the number of mapped reads, and alpha means the signal-to-noise ratio. It can be approximate to flip, a fraction of raising peaks. So N equals number of binding events, in other words, number of peaks, and W is a peaks wise. Based on these findings, we defined BSN, virtual signal-to-noise ratio, as a ChIP-seq quality control metric. So BSN is defined as the equation, and it is equal to the function of N and alpha. So note that here large M and W can be calculated before peak calling, so BSN can be calculated before peak calling. So why BSN can use to evaluate the ChIP-seq quality? So ideally, N and alpha are independent because N number of binding events is dependent on the biological background, and alpha signal-to-noise ratio is basically experimental factor. But however, in actual data, they should correlate because if alpha is increased, we can observe more peaks. So BSN indicates N by double alpha. So in equation, if we assume that N by alpha is some constant, we can use BSN as a parameter to determine the rank of alpha. So for more details, please check our publication. So in this study, we evaluated BSN by using the large-scale public ChIP-seq data. So here we analyze the encoded human ChIP-seq data, and here is a comparison between BSN calculated BSN values and the corresponding value which calculated by peak calling results. And here we use more than 8,000 experiments. So BSN and its expected value is highly concordant. Another advantage of BSN is normality. So here we compare the log transform the flip and log transformed BSN values, and we check the normality of these metrics. So for log transformed BSN, it can be characterized by the normal distribution. So in other words, if we can obtain the BSN values from the public ChIP-seq data, we can obtain the distribution which represents the VSN range. For new experimental data, we can rank them by using the percentile of the non-VSN distribution. So here we introduce an example of the QC workflow by using VSN. So here we assume that we performed an experiment that has two biological replicates. So first of all, we can calculate the VSN values before p-calling, and VSN values can be compared between replicates like ChIP. Next step, if we know the VSN values for public data, we can compare them with our data. So, for example, here is a estimated distribution based on the encoded human ChIP-seq data. So here the replicate we picked up one is a top 10% of the quality, and the other is top 6% quality. So, and another method of utilizing VSN is calculating the expected number of peaks. So, so in our assumption, N and alpha is correlate. In fact, based on the p-calling results, the regression analysis result indicates that N and alpha are correlate. So by using this relation and the VSN value, we can calculate the expected number of peaks by using VSN, and then we perform the p-call analysis. We can compare the expected number of peaks and the actual peak number of peaks. So for these examples, one was a replicate indicates the nearly same number of peaks for expected number of peaks and actual number of peaks, but the other is not so. So in this case, we can expect that former one is failed in the p-calling steps or before, and actually in this case, only 2.2 ml were usable for that sample due to duplicates. Thank you for listening.